السلام عليكم ورحمة الله وبركاته. اليوم هنتناول المحاضرة الثانية من الابيثيليال تيشو. في هذه المحاضرة our objectives will be to identify the specializations of the apical epithelial cell surface, to identify the different types of covering or lining epithelium. The contents of this lecture will be specializations of the apical cell surface, which include microvilli, stereocilia, and cilia, the divisions of epithelium, the different types of covering or lining epithelium. Specializations of the apical cell surface includes three main specializations: microvilli, stereocilia, and cilia. The microvilli are finger-like cytoplasmic projections, and each microvillus contains many bundles of actin filaments, capped and cross-linked by different actin-binding proteins. In this image, on the left side, we have a photo taken by transmission electron microscope for showing the microvilli on the apical surface of the absorptive intestinal cells. On the right side, we have a histological section for the microvilli, showing the microvilli with light microscopy. The microvilli, the apical surface of the intestinal cells, microvilli are dense and uniform, and they form the striated or brush border. They increase surface area for absorption. The stereocilia, they resemble microvilli, and they are typically much longer and less motile than microvilli and may show distal branching. They are found in the epididymis and the proximal part of the ductus deferens. Sometimes they are called immotile, a type of immotile microvilli. They are longer than them, and they show distal branching in the distal part of them. This is the epithelium in the epididymis. On the surface of the epithelium, we have the stereocilia. These stereocilia, they are longer than the microvilli, and they show we have some branching on the distal part. We finished microvilli and the stereocilia. While now we are going to discuss the cilia, which are the long projecting structures, and they are larger than microvilli, larger in diameter and longer, both of them. They contain an internal array of microtubules. We have primary cilium. The primary cilium is a non-motile type of cilium enriched with receptors that detecting light, motion, or odors, or some flow of fluids across the surface of the epithelium. So it contains chemosensors and osmosensors and mechanosensors, and it's found in almost all cell types. But we are going to talk about the motile cilia, which is found only in epithelium, and they are much longer and 2 times wider than the microvilli. This image shows cilia. On the left side, we have in a section, histological section shown by light microscope, showing the cilia. This C stands for cilia, and G for goblet cells, which are mucus secreting. On the right side, we have transmission electron microscopic picture. This picture taken by electron microscope. These are the cilia. These small projections are microvilli, and P stands for basal body. Each cilium has a core structure consisting of 9 peripheral microtubular doublets around 2 central microtubules, which constitute 9 plus 2 assembly. As we see here on the right side, we have image taken by electron microscopy. On the lower corner, left corner, we have a cross section through the cilia, and we notice that in the core of each cilium we have 9 doublets or 9 pairs of microtubules surrounding a central doublet of microtubules. They are forming 9 plus 2 assembly, and this 9 plus 2 assembly is called axoneme, which is attached to the basal body, which is we refer to it by B letter, basal body. C stands for cilia, and MV stands for microvilli. Now we have finished the talking about the apical specializations. Now we are going to discuss the divisions of the epithelium. We have 2 types, covering or lining epithelium, and the second type is secretory or glandular epithelium. In this lecture, we will discuss the covering or lining epithelium. The coming lecture, we will talk about the glandular epithelium. The covering epithelium can be classified according to cell shape into squamous, cuboidal, or columnar. Squamous is a flattened cell shape. Its width larger than its height. The cuboidal cells, its width, height, and thickness or depth are similar. In the columnar cells, we have the height is larger than the width of the cell. According to the number of cell layers, you can classify it into simple epithelium, consisting of single cell layer, 1 cell layer. Stratified epithelium consisting of 2 or more layers. Pseudostratified epithelium. It's still a simple type of epithelium, but it appears as if it is stratified due to the arrangement of the nuclei at different levels. The covering or lining epithelium can be classified into the following: simple, number 1, squamous, cuboidal, or columnar; number 2, pseudostratified; 3, stratified, and the stratified type can be subdivided into stratified squamous keratinized, squamous non-keratinized, stratified cuboidal, stratified columnar, and transitional. So we have simple squamous, simple cuboidal, and simple columnar. So the stratified columnar epithelium, stratified squamous keratinized, stratified squamous non-keratinized, stratified cuboidal, stratified columnar, and stratified transitional epithelium. We will talk about each one by one. The first one is the simple squamous epithelium, which is a single layer of squamous cells. Lining examples for simple squamous, it lines the blood and lymph vessels, and also the serous lining for cavities, pleura, pericardium, and peritoneum. Also, it's found in other regions like lobe of Henle, the kidney. Functions of the simple squamous epithelium is fluid transport, gaseous exchange, lubrication, and reducing friction. In this image on the left side, this is image of histological section taken by or seen by light microscopy, and we see this section of venule, and in the venule we have simply squamous epithelium. These dark structures are the nuclei of the squamous cells. Here, this is a schematic diagram or graph for the simple squamous epithelium, basement membrane, lamina propria, and the glandular tissue, and the capillaries, which are also lined by simple squamous epithelium. Simple cuboidal epithelium, a single layer of cuboidal epithelial cells, is present in thyroid follicles, renal tubules, ducts of exocrine glands, and the surface of ovary. The functions of the simple cuboidal epithelium are secretion, absorption, and surface barrier. This example image for simple cuboidal epithelium, renal collecting tubule, you see this is a longitudinal section, and we have the simple cuboidal epithelium here and in this side. The simple columnar epithelium consists of single layer of columnar epithelial cells. It's present in the stomach, small intestine, colon, and gallbladder. Its functions is transportation, absorption, secretion, and protection. Transportation of substances and molecules. The simple columnar epithelium can be seen in the lining of the gallbladder, as we see here. The cell's height is longer than or larger than the width of the cell, and the nuclei are more basal, is the base of the cells. Now we go to pseudostratified columnar epithelium, which consists of tall irregular cells. All the cells are attached to the basement membrane, but not, and the nuclei are at different levels, but not all the cells extend to the pre-apical surface. Tall irregular cells, all the cells attached to basement membrane, not all of them reach or extend to the pre-apical surface, and the nuclei are found at different levels. Because of that, we say it appears as if it is stratified, but it's not stratified. It's simple. All the cells are attached to the basement membrane. All the cells are attached to the basement membrane, but not all of them reach the surface. Some of them are shorter, some of them are taller, and they reach the apical surface. And we have some cilia here on the apical surface. The pseudostratified columnar epithelium This is an image of pseudostratified columnar epithelium of the upper respiratory tract, either in the nasal cavity or in the trachea. We will see this type of epithelium, ciliated pseudostratified columnar epithelium. It's present in the nasal cavity, trachea, upper respiratory tract in general. The functions: protection, secretion, and cilia-mediated transport of the particles trapped in mucus out of the air passages. So it transports the particles that are trapped in mucus out of the air passages by the help of the cilia. So until now we finished simple squamous, simple cuboidal, simple columnar, and pseudostratified columnar epithelium. Now we are going to discuss stratified squamous keratinized epithelium. Stratified means 2 or 3 more layers. Squamous, the shape of the cells is squamous. Keratinized, the surface layer contains keratin. Epithelium is a tissue. It is present in the epidermis of skin. The functions is protection and prevents water loss. This is the epidermis. This is an H&E stained section of the skin, epidermis of the skin, and we have the superficial layer. There is keratinization, and we have multiple. We can see here 2, 3, more than 2 layers of cells. So this is stratified squamous keratinized epithelium. The stratified squamous non-keratinized epithelium is present in the wet cavities, mouth, esophagus, larynx, vagina, and the anal canal. Its functions is protection, secretion, and prevents water loss. The stratified squamous non-keratinized epithelium, as an example, this image in the esophagus showing the esophageal lining, the longitudinal section through the esophageal wall, and in the lining we see the stratified squamous epithelium, and it is non-keratinized. We can see the nuclei of the cells in the superficial layer. Then we shift to stratified cuboidal epithelium, which is present in the ducts of the sweat glands, in rectal junction, and developing ovarian follicles. The stratified cuboidal epithelium, its functions: protection and secretion. Example of this stratified cuboidal epithelium is the excretory ducts of sweat glands, as we see here. Is an H&E stained histological section of sweat glands, and these are the excretory ducts of sweat glands. The stratified columnar epithelium, present in conjunctiva, large excretory ducts of exocrine glands, and parts of male urethra. Also, the stratified cuboidal and stratified columnar epithelium, they are rare and they are found in the excretory ducts of some exocrine glands. The stratified columnar epithelium functions: protection, absorption, and secretion. As we see, stratified columnar epithelium is present in the conjunctiva of the eyelids, large excretory ducts of exocrine glands, and some regions or parts of the male urethra. Example of stratified columnar epithelium is the duct. This is the large excretory duct in salivary gland, interlobar duct. And we see there is 2 layers. At least we can see 2 layers of columnar cells. The last type we will talk about is the transitional epithelium, which is a form of stratified epithelium, highly specialized to accommodate a great degree of stretch, and so named because it has some features which are intermediate, transitional between stratified cuboidal and stratified squamous epithelium. It is characterized by the presence of superficial layer of large dome-like cells called umbrella cells. These umbrella cells are specialized to protect the underlying tissues from the hypertonic and potentially toxic effects, cytotoxic effects of urine. And it is found in the urinary tract from the renal calyces, ureters, and the urinary bladder. As we see here in this drawing or schematic drawing, we see the basement membrane, basal epithelium, and on the surface we have the umbrella cells. On the right side, this is a histological section, H&E stained section seen by light microscopy, and we see the superficial cells are dome-shaped, dome-like cells called umbrella cells. This transitional epithelium is found in the urinary bladder, ureters, and renal calyces. Transitional epithelium is related to the urinary system, urinary tract, urinary bladder, ureters, and renal calyces. In the urinary bladder, when the bladder is full, these layers will be compressed and distensed. The epithelium has the ability to be, the epithelium is distensible, has the ability to be distended. And the layers, when the bladder is full, the wall of the bladder will be stretched and the number of layers will be about 2 to 3. When the bladder is empty, so that the bladder is empty and relaxed, the wall of the bladder and the transitional epithelium appears as 4 to 5 layers. So it has the distensibility property. The function of the transitional epithelium is the protection. As we see, the umbrella cells will protect the underlying tissue from cytotoxic and potential cytotoxic effects of urine and protects the underlying tissue from the hypertonic urine and cytotoxic urine effects of urine. Functions: protection, as we said, and distensibility. It has the ability to be distanced. When the urinary bladder is full, it appears as 2 to 3 layers of cells, but when the bladder is empty, it will appear as 4 to 5 layers of cells. Now we finish the types of epithelium. We are going to summarize what we have talked about. There are 3 main types of apical surface specializations: microvilli, stereocilia, and cilia. The epithelium can be divided into covering, lining epithelium, and glandular epithelium. In this lecture, we covered and finished the lining epithelium or covering epithelium. In the coming lecture, we will talk about glandular epithelium. The covering epithelium can be classified according to the cell shape and the number of the cell layers